Once again, welcome back. We're going to be going over proteins in this fourth video on Chapter 3 material. So proteins are one of the most abundant macromolecules in living things. They're also the most diverse in their function compared to the other macromolecules. So here are some examples of their functions. First, they can regulate the cell, both in function and in their structure. Interestingly, proteins can also protect cells and even protect whole organisms through the generation of some kind of immune response, or even through the formation of protective structures outside of the cell membrane. Proteins can form channels that allow the transportation of things into and out of cells. And many enzymes, actually most enzymes, are composed of proteins, and these speed up reactions. Finally, even some of our toxins are proteins, such as toxins released by certain bacteria that cause tetanus or botulism. As we learned earlier, enzymes are molecules that speed up or catalyze reactions. So catalyze just means speed up. So because they speed up reactions, we say that they are catalysts for these reactions. And there are many different types of enzymes. Catabolic enzymes break things down. Anabolic enzymes build molecules. They build larger molecules. And catalytic are things that speed the reaction rate. But all of these will actually speed up the reaction rate. Usually enzymes will. Here's an example of an enzyme that happens to be a protein or comprised of proteins. They are very specific in what they will bind to as their substrate to cause or speed up the reaction. And this table is nice because it gives us more examples of what proteins can do. What are their functions? They can digest, so digest the food that we eat. Amylase, for example, we talked about before, digests carbohydrates. Lipase, lipids, and then pepsin and trypsin digest proteins. Transportation, I talked about moving things into and out of the cell, but we can even move things around through the blood, through these proteins. For example, hemoglobin allows us to transport oxygen from the lungs into our muscle area, our muscle tissues, and they deliver oxygen there when necessary. Structural proteins can determine the shape of the cell as well as different types of body parts. Keratin, for example, makes up our hair and our nails. Hormones, so proteins are important in terms of hormone production as well. Insulin is a type of protein hormone, as is thyroxine, a type of hormone that determines our metabolic rate. I mentioned earlier, proteins are important in our immune system and defense. For example, immunoglobulins can bind to foreign substances and prevent them from invading further. We have proteins called actin and myosin that are involved in muscle contraction, and we can also store amino acids and other nutrients in protein format. Egg white, for example, or it's actually called albumin. Albumin is egg white, is a way for us to store proteins, and we can store proteins in terms of protecting the embryo when we're looking at plants and seeds. Just as monosaccharides, or the subunits or monomers of our complex carbohydrates, amino acids are the monomers or subunits of our proteins. There are about 20 common amino acids that are found in proteins, and we see a generic picture of an amino acid here. So all amino acids will have a central carbon, sometimes it's called an alpha carbon. Almost always depicted on the left side, we have the amino group. On the right side, we generally have it on the right, is the carboxyl group. And then we have hydrogen also attached to the central carbon. The difference between the 20 amino acids we have really lies in the type of side chain an amino acid has. And I would like you to be able to sketch a generic amino acid like this one on our exam. So if I asked you to draw an amino acid, I'd like you to be able to draw a central carbon, and you know that there are 4 things attached. You don't have to draw it like this model, but you want to be able to write that there's an amino group on one side, a carboxyl group on the other, a hydrogen attached to that central carbon, and that side chain or R group is what varies between the 20 amino acids. So of those 20 amino acids, 9 of them are essential, which means we can't make them ourselves as humans. We need to get them from someone else, and we do that through our diet, the food we eat. Of those 20 amino acids, they differ in their R group or side chain. Sometimes it's also known as a variant group, although I don't really see this term very frequently. We have several types or classifications of R groups. Some of these R groups are nonpolar aliphatic, some of them are polar, etc. And we're going to see this in the next slide. Here we can see the different amino acids and how they vary by their side chain, that R group, which is highlighted in blue, as we can see throughout the whole figure. They're grouped into different categories based on the side chain's characteristics or properties. I can see these are nonpolar aliphatic R groups, aliphatic meaning that they're in chain form. The side chain is in a chain form rather in a ring form. Nonpolar again meaning that it does not like water, so insoluble in water. We have polar that are uncharged. We have positively and negatively charged side groups, and then nonpolar that are aromatic in ring form. Looking at this, you do not have to memorize each amino acid, although you'll see the names of several of these pop up fairly frequently throughout the semester. You don't have to memorize, for example, that lysine looks like this, or be able to sketch it out. But I would like you to be able to recognize whether or not something is going to be hydrophilic or hydrophobic. So, for example, let's say I gave you a picture of isoleucine on an exam, and I asked you, based on this picture and looking at this side chain, do you think the side chain is polar or nonpolar? And I can see that I don't see anything like an OH or an NH. So I would say, oh, it's hydrocarbons. So I would say, oh, this is nonpolar. It's going to be a hydrophobic R group or side chain. Anything that's charged or polar will be hydrophilic or water-loving. Why is this important? Why is it important to know which side chains are water-loving, which are water-faring? Because this can help us predict what a protein might look like and which amino acids will be facing in what direction if they are placed in water. And your book poses an interesting set of questions in this chapter. Let's say I have a soluble protein, so it's soluble in water. Let's say I have some water here, and then I put a protein in here, so there's some kind of protein, three-dimensional shape. So what kind of side chains would I expect on the surface? So if the surface, let me change colors, if the surface is facing the water, I would say, oh, the R groups that are facing the surface where the water is will probably be charged or polar. What about on the interior? Let's see what other colors I have, purple. So anything in the interior is probably purposely facing away from the water because they don't like water. So it could be anything that is nonpolar. And then the second question, or the end down here, they say, what would you expect to find in a protein in a lipid bilayer? Let's see. Do you guys remember that phospholipid bilayer we saw earlier when we were talking about lipids? We have our polar heads that are the circles and our non-polar tails. So if I had a protein embedded in here, so it's some kind of protein like right there, that green thing, then I would say that any part of the protein that is in that hydrophilic region, those R groups are probably hydrophilic. And anything that is Here, so the protein, the R groups that are part of the protein portion in this orange color, those are probably hydrophobic R groups found there. Going back to those 20 types of amino acids that are commonly found in proteins, we said that 9 of them were essential. Essential means we cannot make them, humans cannot produce them, so we need to eat them. We need to get them from our diet. This is similar to when we were talking about lipids earlier. We saw that there were a few fatty acids that were essential, like the omega-3 and omega-6 fatty acids, where we could not make them, so we had to get them from our diet. If you are a meat eater, most of the meat or animal proteins will have all of them. But if you are vegetarian or vegan, most of our plant protein sources don't have all of these. So you have to mix and match your protein sources to make sure you're getting all of the essential amino acids. Non-essential means that we can make them, or we can convert from one of them to a different one within this list, so we don't need to get them from our diet. We're going to see more abbreviations towards the end of the semester, but I wanted to let you know that besides the full name of an amino acid, sometimes we'll see them abbreviated either by a single uppercase letter or by three letters. So valine can be represented by capital V, or VAL, aspartic acid, by the letter D or ASP. And again, we're going to see these again later on. You do not have to memorize this. What's important here is that the sequence and number of amino acids is very important in determining the shape of the protein, the size, and the function of the protein. Okay. So how do we make proteins from amino acids? You remember going back to our talk on carbohydrates? Glycosidic bonds were the type of covalent bond that linked monosaccharides to form polysaccharides. Similarly, for amino acids, in order for those to form proteins, we need to connect them. And the bond type that is found here are called peptide bonds. So peptide bonds are a type of covalent bond that link amino acids in order to form polypeptides or proteins. So here we have 2 amino acid monomers, and what we're going to see is that this is going to go through a dehydration synthesis reaction where water comes out of the reaction, and you're going to form a peptide bond. If we had another amino acid coming in, so amino acid coming in over here, we would see that the carboxyl group of the previous amino acid of the chain will be linked to the amino group. The amino group would be over here somewhere on the left side of this. So these two would be linked to form the next peptide bond. And again, another molecule of water would be released. If you have 2 amino acids linked together by a peptide bond, so let's say I have 2, amino acid 1, amino acid 2, we call this a dipeptide. It's a really short molecule. If we have 3, so if we had 1, 2, and the third one linked together, we call this a tripeptide. Tripeptide, dipeptide. And if I look really closely, I can see the backbone of my little chain is NCC, NCC repeating, nitrogen, carbon, carbon, nitrogen, carbon, carbon, and so forth. And almost always, again, you see the amino group written on the left of the chain and the carboxyl group written on the right side of the chain. We often use the terms polypeptides and proteins interchangeably, but technically they're different. Polypeptides are a bunch of amino acids linked together through those peptide bonds. In order to be functional proteins, however, usually more things have to happen. So a protein, yes, is a polypeptide, but usually there are other things that will be attached to the polypeptide chain or chains before it becomes functional. These polypeptides also have to go through a folding process to create a 3-dimensional shape that allows their proper function. And many of these polypeptides, once they fold up properly, They actually have to go through further modification before they can be active and catalyze reactions or perform their proper function. As I mentioned earlier, a protein's shape is very important to their function. For example, let's say this yellow structure is my enzyme, and it binds to a substrate to speed up some kind of reaction. The area where the substrate binds is called the active site of the enzyme. And if you were to change the shape of this active site even slightly, then this enzyme would no longer be able to recognize its substrate or bind to it or cause the normal reaction. How do proteins get their final shape? There are actually 4 levels of structure that the protein has to go through. We call them primary, secondary, tertiary, and quaternary structure, and they're often abbreviated with these symbols. So the first level of structure is primary protein structure, and what this is the unique sequence of amino acids that's found in the polypeptide. We usually see this as a linear chain. A protein can have multiple chains in its primary structure, and we see this in an example provided by the book. The hormone insulin, which regulates blood sugar, has multiple chains. So the main bond that's important here are the peptide bonds between the amino acids. And remember, that was a type of covalent bond, that peptide bond. The order of amino acids is very important. If you were to delete an amino acid or even change the order of 2 random amino acids, you might get a completely non-functional protein, or you might get a completely different protein. So how does our body know what order to place those amino acids in to build a proper protein? It comes from our genes, our DNA. So if there's something wrong with the DNA, if we have a mutated gene, for example, this could lead to mutations in the protein structure, leading it to be non-functional or have irregular function. Earlier, I mentioned one of the functions of proteins is transport, and I used hemoglobin as an example of a protein that transports oxygen from our lungs to our muscle tissues. In normal hemoglobin, one of the protein chains or polypeptide chains has this sequence of amino acids, but there is a genetic change that happens in some individuals where one single amino acid is different. And I can see it's amino acid number 6. Glutamic acid is replaced by valine. Just that single amino acid chain causes something called sickle cell anemia. With this valine, the hemoglobin changes shape, and that causes the whole red blood cell to change shape and not be able to perform their normal function. The blood of individuals with sickle cell anemia can be smeared on a glass slide and examined under the microscope, and you can see sickle cells have this flat crescent shape or moon shape instead of the nice globular shape of healthy red blood cells. So individuals can have 1 copy of the sickle cell gene or 2 copies, and those with 2 copies have very serious health problems. Those with 1 copy, sometimes they can't tell they have sickle cell anemia until they exert themselves if they try to exercise. Symptoms include things like breathlessness, dizziness, a lot of pain, headaches. And we're going to learn more about this when we get to genetics. This is exam 3 material. So primary structure was the linear sequence of amino acids formed by those peptide bonds. As soon as that's formed, these polypeptides start folding upon themselves a little bit, and that leads or takes us to secondary structure. So secondary structure is due to the formation of hydrogen bonds on the backbone of the polypeptide chain. So we have 2 types of secondary structures that you'll see in these polypeptides. Alpha helix or alpha helices, plural, look kind of like this. And again, these are due to hydrogen bonds. Beta-pleated sheets are also formed as a result of hydrogen bonds, and they look kind of like this, and you'll see them in the next picture. Again, secondary structure is a result of hydrogen bonds formed through the peptide or polypeptide backbone. And it depends on whether or not you see these or what combination you see really depends on the type of protein you have. This is a nice picture of that tertiary structure where you really start seeing that three-dimensional folding or three-dimensional shape. And we see some hydrophobic interactions between two nonpolar side chains. We see some ionic interactions. I see a hydrogen bond over here and that covalent linkage I showed you earlier, the disulfide bond. And tertiary structure always reminds me of hair. I wonder how your hair is in the morning, because my hair, whenever I wake up every morning, I look like a crazy woman. I don't know how people wake up with like perfect hair. And I want to think about I know hair is protein, right? Hair is made of keratin protein, and it doesn't always look the way I want it to. So if I wake up in the morning with messy hair, what are things that I can do to fix my hair? So some things I do is maybe I just smudge some water on it if I'm in a rush. Maybe we use hair gel, maybe use a flat iron or a blow dryer to make your hair the shape you want. So when we're doing things like that, we're breaking the weaker bonds. So we can break the weaker bonds, like the ionic, the hydrogen bonds, those hydrophobic interactions. We can break the ones that formed overnight due to pressure, like sleeping on one side of your head, and then reform them in the shape that we want using the hair gel or the blow dryer. But permanent changes, like if you have straight hair but you want curly hair or vice versa, require permanent changes, covalent changes, and that needs chemical treatment. If you have very curly hair, like the folks shown here, that means you have more disulfide bridges, more of these disulfide bonds in your keratin protein. But if you have very straight hair, you have fewer of these disulfide bonds in your hair. All right, our last level of protein structure is quaternary structure. Not all proteins have quaternary structure. You only see quaternary structure when a protein is made of more than one polypeptide, So more than one of those chains of amino acids. To form quaternary structure, those multiple chains of amino acids can interact through all four bond types as well, just like they did in tertiary structure. They can interact through hydrophobic, ionic, hydrogen bonds, or even those covalent bonds. This is a really nice figure to summarize those four levels of protein structure. We had primary structure, that linear sequence of amino acids created by the formation of the peptide bonds between the amino acids. We have our secondary structure, which is formed by hydrogen bonds in the backbone. We have our tertiary structure, which is a 3D shape formed by interactions between those R groups, and you can see all four bond types. And then quaternary structure is when you have more than one chain of amino acids and they connect. Notice that even though this one has 4 polypeptides that create quaternary structure, it doesn't have to be 4. It could be 2 polypeptides coming together. It could be 60 polypeptides that come together. The thing about protein structure in that three-dimensional shape, it can be changed if the interactions between those R groups or between those polypeptide chains are broken. And how can you break them? So you can take a protein and move it to a different pH or change the temperature of the environment. Those are probably the two most common ways. Changing the shape or unfolding or losing that three-dimensional shape, we call that denaturation. Changing the shape of the protein is usually detrimental to that protein because they can no longer function properly. In some cases, it will be reversible as long as that primary sequence of amino acids is still there, but in some cases, it's not reversible. So when I was in college, I don't know why, but my roommate and her friends used to do this thing where they would test our teaching assistants to see if they were, quote, smart or not. And the question they would ask their TAs is like, hey, how come most things melt? They go from solid to liquid when you heat them up, like ice cubes or chocolate. But why is it, weirdly enough, that when you heat up an egg, it goes the opposite direction? It goes from liquid to solid. And if the TA knew the answer, then they would deem that TA smart, which I don't think is necessarily true. But why is this? Why does this go in the opposite direction instead of what we see on the left? This phase change right here. You're probably already thinking it, but because we know that eggs are comprised of protein, the white part of the egg is egg albumin, a type of protein, and when we heat up the albumin, the egg white, then it denatures, those proteins denature, they unfold, and it goes from liquid to solid. So what we see is this globular or three-dimensional albumin protein is at first in liquid state, but as you heat it up, it unfolds, it linearizes, loses its shape. At the same time, the egg, the shell of the egg is actually not waterproof, so water will leave, and these proteins condense to form that solid final structure of the cooked egg. That takes us to the end of the fourth video, and in the next and final video for this chapter, we're going to be looking at nucleic acids, and that is our fourth and final type of macromolecule.